everybody. My name's Helen Groves. I'm a person living with Parkinson's. I've had Parkinson's just over 6 years, or I've been diagnosed just over 6 years. I took early retirement from work due to Parkinson's. I was a pharmacist, worked in community pharmacy and also in NHS commissioning. So I have big interest in research and medicines in general. Was a participation in the exenatide trial. I took the placebo, so I injected myself for 96 weeks with salt water. But obviously very disappointed in the results of that. But as I keep saying, the fight must go on and we have to keep going. Thank you.
And Tim, if I could ask you just to introduce yourself as well, please.
Hi. I'm terribly sorry to have technology failure. Hi, I'm Tim Brasher. I've had Parkinson's for 20 years. We don't have a family history or anything like that. I've been involved in 3 clinical trials and various other paperwork-based trials. What else can I say? Thank you.
Tim, can I just ask you, before we have questions from the audience, you said you were involved with 3 trials. What was the initiating factor for you with regards to getting involved in clinical trials? This is an inhibiting step for a lot of people sometimes.
Yeah, I wanted to be at the front of what was currently available. I'm calling it selfish, but if there's a cure being made, I want to be at the front of the queue to get the real deal. Yeah, I'd recommend clinical trials. They're, you meet a nice bunch of people, you learn about your own condition, and you get a free health check every few months.
Indeed. And Helen, is this similar for you?
Yeah, I mean, obviously in my line of work, research is really important. And when I was diagnosed, I think I was one of the few people who had a reasonable diagnosis as it can be. And the consultant that diagnosed me said, are you interested in research? Because if you are, I'm going to refer you to Professor Fultonay at Queen's Square because he's doing this research into exenatide. And it was just a no-brainer for me. I mean, I didn't know you got so well looked after, and that's definitely a plus. You get lots and lots of tests and opportunities to speak to clinicians and get your meds adjusted, etc. But for me personally, it's kind of like a giving back thing. And it's just knowing that you're doing your bit so the next generation of people that get the same horrible news that we got maybe will face a more promising future. I mean, I think probably for me, or for people that already got it, a cure is probably, I think at best that would stop it. But it'd be really nice to know in the future, and that's a kind of just nice to know that you're involved in that you're actually going to be part of it, and that you have done your bit towards it.
Wonderful. Okay. Any questions from the audience?
Yep. You can hear me?
Yep.
Yeah. My name is Lydia Lonergan, and I think this question is more for Simon. The multi-arm, multi-thingy-bob trial.
Multi-stage, yeah.
Multi-stage. So every trial you hear about, you have different requirements that are applicable to join it.
Eligibility criteria.
Yes. So when you have the multi-arm, multi-stage, how do you know that the people you've selected will be then applicable as you go through dropping one study in and out all the time?
So I will ask Georgie and Millie to correct me here if I'm wrong, but the ASPro-PD platform's got a very broad eligibility criteria. It's going to be taking all comers, you could say. The age range is from 30 to 80, and is there a limit in years since diagnosis?
To my knowledge, no. Not for
no. This is a good memory test for all of us. Yeah, it's very much all comers.
So that, for example, would mean that if the Ambroxol had been part of that multi-arm, it wouldn't be able to work because it's only people under 7 years. Do you see what I mean?
Yes, but the Ambroxol study is specifically designed to test a particular hypothesis.
I understand that.
Yeah.
Thank you.
Hi, my name is Vipa Telly. I'm here because a member of my family has recently been diagnosed with Parkinson's. And thank you all for an amazing presentation. My first question, I have two questions, sorry. One was on Ambroxol. You mentioned the mucolytic activity, breaking down the mucus. Are there other agents that you'd be looking in the future that we use currently as mucolytics in the NHS?
So maybe I can answer to this one. I think that the effect that Ambroxol has on the GBA pathway and on Parkinson's is not related directly to the fact that it's also a mucolytic agent. It's due to the fact that it binds to GBA and helps it to fold properly. So this is specific to Ambroxol. So as far as we know, there are no grounds to suspect that other mucolytic agents might have a similar effect on Parkinson's.
Okay, thank you. And the second question was, do you think there's any evidence that the COVID vaccine may have contributed to increased cases of Parkinson's disease? Because I think Millie mentioned you increased the recruitment, so that makes me kind of think, have there been more cases? Has anybody looked if the cases have gone up since people had the vaccine, since 2020, as we're 5 years down the line? Is there a trend that there might be a link to the vaccine? Just putting it out there.
They've done epidemiological studies looking at large nationwide cohorts, looking at medical registry data, and the evidence suggests that Parkinson's is one of the fastest growing conditions. It's growing at a faster rate than simply the aging population in Western cultures. And very shortly, Parkinson's UK, I believe, are going to be putting out a new report on the statistics of Parkinson's, the incidence within the UK. So that'll be something to look out for.
Okay. Thank you.
Hello, Rory Kethling-Jones. I was impressed, but also almost horrified by, I think it was Mairead's presentation, about the sheer number of steps to be gone through in this Ambroxol trial. We're talking about speeding up trials. I get the impression that there's ever more bureaucracy. This is not a complicated medicine, is it? This is basic cough medicine, and you seem to be forced to go through so many hoops. It seems to take so long to recruit people. Are we really advancing in the way we carry out these trials?
Thank you for the question, Rory Kethling-Jones from the Movers and Shakers podcast. Mairead, this is your opportunity to rant about the frustrations and all the steps of setting up a clinical trial.
Yeah, I mean, I agree. It's a lot to do, and it is quite frustrating. You'd think by now we would have a quicker pathway, but I guess in the UK, clinical trials are very safe. They're renowned worldwide because they're done well, and I think a lot of that is because we put a lot of effort into setting them up properly. But I do share your frustration. It takes a long time.
Hi. Carrying on a little bit from the previous question, also, without wanting to flog a dead horse, so the more we increase the exclusion criteria, aren't we going to slow people going on the trial? And so things like DBS being an exclusion criteria, the more people are beginning to have DBS, isn't your recruitment group going to shrink?
Shall I?
Please, Marco.
Yeah. So I think that, of course, it's a tricky question, and it's a very appropriate one as well. I think that the short answer is that when you do a clinical trial, you need to be able to assess whether a molecule, a medication is effective. And people that have a type of treatment like DBS, which is very dynamic, you know very well that changing the parameters can be changed from one visit to the other. makes it very difficult to assess the efficacy of a compound that we are studying. So that's the main answer. Of course, it's not the perfect solution, but it's the best one we were able to come up so far to unfortunately exclude people with DBS from some clinical trials. That doesn't mean that these people will not benefit from a successful trial, of course. If the medication is proven effective, there is no reason why it shouldn't be effective also in people that have DBS. So it's mainly a practical reason that forces us to exclude some people from trials.
Oh, that DBS is not an exclusion criteria for every trial. There are trials that do allow people with DBS to be involved. There is, however, evidence that one of the issues with, one of the complications with DBS is that the assumption is that it's not having an effect on the brain, just stimulating the brain. But there is evidence now to suggest that the Parkinsonian brain is slightly different in a DBS person with DBS than before they had DBS. There's evidence to suggest that DBS could even be disease modifying in a very small way. So that is a complicating factor that they want to remove from the clinical trial by having DBS as an exclusionary. But it's not every trial. So there are some trials that do allow DBS to be involved.
The shrinking of the group that's eligible, because
Yep.
so many more people are having, you know, you go to the consultant and they say, well, we could do this, there's no trial available, you could have DBS. Well, the more people that have them, and when you exclude it, aren't you then reducing your potential sample size and then slowing the process down even further?
Yep, agreed. Where are we going?
Hi, Jeff Boxer. Is that on? Hello? Yeah.
Yes.
Sorry, Jeff Boxer here. Just a scientific question about the GBA variants versus the idiopathic cases. Is there any sort of, what's the link in terms of response to various treatments or progression of disease between those GBA variants and the idiopathic cases? Is there any evidence from that different people will do better on different regimes and different...
Yeah, so there is some knowledge that people with Parkinson's that have a GBA variant have a slightly more severe phenotype, so a more severe form of Parkinson's, with slightly more frequent non-motor symptoms, like, for example, cognitive impairment, although the differences are relatively small. And among GBA variants, the ones that are considered more severe, we define them severe in association with the other condition, Gaucher disease, they also are associated with a slightly more severe form of Parkinson's compared to the carriers of mild variants. That being said, to my knowledge, and Professor Shapira will correct me if I'm wrong, there is no evidence so far that people with severe variants will benefit from Ambroxol more than carriers of mild variants, for example. And also I wanted to add, because I don't think it's been mentioned, that the hypothesis of ASPro, of the trial we talked about, is that even people that have Parkinson's but don't have a variant in GBA might benefit from Ambroxol, because the pathway of GBA is affected in all people with Parkinson's, not only those that have a GBA variant.
In the second row here.
Hi, God, that was closer than I realised. Claire Lehman, young person with Parkinson's, diagnosed at the age of 37, had it for almost 8 years. So mine's a kind of question about urgency. And firstly, thank you for the meeting this afternoon, and thank you also for such brave and courageous steps in how we're doing trials differently in a more efficient way. I was just thinking about the GLP-1s. And I'm really heartened to hear about the fact that after the latest trial comes up, there's going to be a wash up and look at those things. And I'm also thinking about those contradictory results with those two trials, for exenatide and exenatide, and potential how this might look in terms of Ambroxol. When we're looking at the effectiveness of interventions, and we define those primary endpoints before we start our trial, do us as scientists, do we as scientists think sufficiently about if it doesn't come, do we keep such an open mind for those defining those endpoints, and also do we also include other measures, for example, patient-reported outcome measures, in a consistent fashion? And could we do that such that when future trials come to light and other things, that we could then aggregate those retrospectively to better interrogate our data? So it's quite a long-winded question, but.
So is it the difference between clinically meaningful endpoints and patient meaningful endpoints?
A bit of both, and also that flexibility such that we're able to almost have a bit of a consensus statement about how we define our endpoints before we going forward with all trials, such that there was an international approach so that we could interrogate that big data more effectively going forward.
Everybody's looking at me. So
Who would like that one?
we spent an awful lot of time discussing about the endpoints for ASPro, and as previously, there is no perfect outcome, no perfect endpoint. We want to, of course, the final result must be that people feel better, because otherwise, if we see that a blood test improves but people feel worse, there's no point in the treatment. So that being said, you also need to have measurable and objective observations, because we know that up to 25% that every response to medication is placebo, so it's linked to psychological reaction. So there's a lot of different things to keep in mind. In the end, for ASPro, we decided to use the UPDRS, which is made up of sort of an objective examination from the clinician and a questionnaire that comes from the patient. They're merged together into one outcome, and this is the primary outcome. Then we have a number of secondary outcomes, which include and Mairead will correct me, but include questionnaires about quality of life, non-motor symptoms, severity of the disease as perceived by the patient. So I think that, and cognitive outcomes and other things. So again, there is a hierarchical order in these outcomes, but we did try to include as much as possible both objective, subjective, clinician and patient views into the outcomes.
Helen, can I come to you with this question? Mm-hmm. Having taken part in a clinical trial, you were asked to do the UPDRS clinician-based assessments. What were your thoughts following your experience of a clinical trial with regards to the more patient-focused endpoints?
Yeah, so the patient-focused endpoints were the various questionnaires that anybody that's done anything online will have done similar. And I think we were clear that the primary endpoint was the UK PDS. But I think following that, they are studying that a bit more, and I know one of the researchers there is looking into that in greater depth. I think one of the most frustrating, slightly diverting on from your question.
Please.
One of the most frustrating parts, I would imagine, for research in Parkinson's is the lack of any sort of specific biomarker, never mind a specific biomarker that actually represents a person's Parkinson's specifically. It kind of reminds me a bit of like now where, we say we treat, if you're doing an x-ray on somebody's knee, like a few years ago, the x-ray would determine what you were doing, whereas now we kind of say we treat the person, not the x-ray. And I think until probably in Parkinson's we get some sort of reliable biomarker, we're going to have this question all the time, because the more you talk about just how much of that is the placebo effect. Because we had people in the exenatide trial who were convinced they were having exenatide, and they weren't. They had, so there's a huge, and if I don't know if most of my readings about Parkinson's, but a lot of stuff I read say that the placebo effect is very big and is sort of big in Parkinson's much more than other things, that we are very prone to that. I don't particularly know why. But yeah, it's a big question. But at the end of the day, what really counts is how somebody feels and how somebody progresses, and how they're walking, and you know, are they able to work. What really matters is how you get on with your life, how your quality of your life is. That is ultimately it. But how do you measure it? It's kind of the holy grail, really.
And Tim, can I ask you, the wearable technology is becoming an important endpoint in clinical trials now. Have you ever taken part in a study where you were required to wear a device or use a device?
Simple answers, no. Sorry. I've seen the devices and got briefly involved with assessing one of the units, a watch type thing. Yeah, that was done through Cure Parkinson's. But it didn't seem to go anywhere. But going back to Helen's talk,
yep.
After about 3 months, there's several people I knew on the trial. We all agreed that we were on the placebo, since we knew what was going on. So at the end, when we got the results, yeah, it was confirmed that we were guessing right, but yeah, it's just a difficult thing to do.
It's a very human thing. It's a very human part of the clinical trial process, I think.
Yeah.
We're always asking, I wonder if I'm on treatment or if I'm on placebo. Question in the middle there?
Hi, my name's Steve Lloyd. This may sound like a really silly question, but is there a component within Parkinson's that could be related to gender rather than brain? Because brain should be the same, but is there something in there that might be different for male and female? Is that something you ever look at, or is it just brains are brains?
Gender.
Gender.
Oh yes, absolutely. I mean, I guess if we talk genetics, we can safely talk about sex. There are differences. The most important thing is that Parkinson's is more frequent in males than females. So around 2/3 of people with Parkinson's are males.
Not in Australia, they've just done some prevalence data, so.
Which is extremely interesting.
Yeah, exactly.
But
not everywhere.
yeah, so in Australia there is a different distribution of male and female, but worldwide this is the most common observation. So that by itself suggests that there must be some biological difference there. We also know in our studies on GBA, we noticed that the carriers of some types of mutations are more frequent, that have Parkinson's are more frequently females than males. We are about to write up this particular finding. And so definitely there are some differences, but that is true in basically everything related to medicine, I would say. I would say that females probably have been neglected a bit by research in the past. And the response to medications is significantly different according to sex.
Okay, because I hadn't noticed that in any of the research that it was male,
There's
female.
just about to be a huge effort to get underway, being led by the Critical Path for Parkinson's, which is going to be collating data around, particularly from clinical trials, looking at gender and sex differences, because we know that this is a big gap. So there's a huge effort to make sure that this analysis happens. And actually for the last 5-6 years, we've been very much insisting that every piece of data that we, for any of our projects, is analysed on that basis as well, because we feel it's hugely important.
And you were suggesting that Australia has recently had some data?
Yeah, they had some data where it was much closer to 50-50, and we know that in Japan that it's inverted. So I think it's not a clear picture that it's more men everywhere in the world, and I think the more prevalence data that we can analyse, then I think we'll get a better picture of what's going on here. But I think it's absolutely essential that we look at at every metric that we possibly can in every clinical trial to learn as much as we can.
As a proud New Zealander, I'd probably suggest that Australia should be treated as an outlier. Questions from online?
Second. Okay, cool. So we have just a few questions from online, if that's okay. The first one is just, you guys mentioned that the dose for Ambroxol changed from the phase 2 to the phase 3. So just sort of what the justification is for that.
The dose we decided to use?
Yeah, was it the dose or was it the, because I know the people on the phase 2 took 21
The
pills.
number of pills, maybe, as opposed to the
Oh
dose.
yes, the final dose in terms of milligrams of Ambroxol is exactly the same. This time we designed the tablets to contain the right amount. Previously we were using commercially available tablets meant to treat chest infections, and so participants had to take a significant number of tablets per day. I remember it was, I think, 30?
21.
21, yes. So that hopefully is an improvement.
Yeah, definitely, yeah. It was an effort.
Yes. But the final dose of Ambroxol is the same. So.
Yeah, it's good.
21 pills a day. Did you have room for food?
No, that's why I'm the shape I am.
Another question from online?
3 lots, there's 7, and they didn't taste very nice.
I'm told the taste has also improved.
Yes. So you got a winner.
Yes.
Another question from online.
Just sort of kind of similarly talking about the medicine, as we know, Ambroxol is approved as a cough medicine in much of Europe, just sort of what the difference is between what is being tested in Parkinson's versus what you can sort of buy off of a shelf.
Well, the first one is the dose. Again, people that want to use the same dose that we are testing, buying it over the counter would have to buy a spoon as well, and but also we would not recommend people to test medications that are not for their indication without medical supervision in the setting of a clinical trial. It's still research, so even though we stress how safe this medication is, we still need some precautions, of course, and we do the trial to figure out whether it's effective, but also whether there are any potential risks. And so it's very important to do it in a very controlled and structured setting, I think.
Another question from the audience. In the second row.
Just thinking about the timeline for recruitment for ASPro-PD, if it looks like you're not getting close to your targets for the right number, will you extend the diagnostic criteria in terms of years since diagnosis? And at which point would you make that call? Is there yeah.
So perhaps Mairead can be a bit more precise. I think that at the moment through PD Frontline we had a massive response, so we're not expecting to struggle to recruit for the figures we are hoping to achieve.
Yeah, I would agree.
But of course, if worse comes to worse, we might have to review some of the inclusion criteria. It would be a very extreme thing to do. It usually doesn't happen once the protocol has been approved. Only minor changes can be made.
And as Amelia and Georgie were suggesting, there's other clinical trial options as well available. Question from the second row to the back there.
Hi, just wanted to ask a question about the Ambroxol. So, during your choosing of participants, will you be choosing a diverse? Do you have a quota of women, people from a diverse background?
So how will you be handling the diversity question regarding the recruitment for the Ambroxol study?
I can probably answer that. So it's a difficult one because we we aren't in control of the actual recruitment, but we make sure to advise all of our sites to approach as much of a diverse participant population as possible. And on the database, we are collecting lots and lots of different variables, and we will monitor that during our interim analysis, just to make sure that everything is balanced as much as possible. And if not, then maybe reach out to the sites and ask them to look at their population and see if they can increase diversity there.
Yeah, and I think Georgie and Millie can also comment on the PD Frontline, I think.
From our perspective, we send across all information for all eligible participants for each site, so it's not that we hit a certain number and stop passing on people's information. You know, we still continue to pass information on, so anybody from any site that is eligible, fitting the preliminary criteria, we send them on, so no one's kind of been excluded in terms of that. And also, we are looking to improve our diversity of our cohort as well, on PD Frontline just as a study itself, regardless of our Broxel. Which is why we're kind of approaching people to become PD Frontline ambassadors in certain communities and certain groups, so that we can kind of spread the word a little bit more about the research that we're doing and the genetic testing and the opportunities that are available for people with Parkinson's who want to look into clinical trials. It's always better to get a wide group of people with, you know, a lot of differences, just simply in terms of research, regardless of, you know, it'd be best if we could get everybody involved kind of thing. So from our perspective, we kind of won't be excluding anybody, we'll be sending everybody on, and we're trying to kind of reach out to as many people as possible to get them involved, with your help, hopefully, as well.
Emily, with the wonderful selection of studies and trials you presented, is this becoming more of a feature that the investigators are looking for, that diversity as well?
Yeah, I would say so, definitely.
Well, GP2, one of the core...
Yeah, but GP2 is a perfect example, exactly.
Yeah, one of the core missions of GP2 is to make research of Parkinson's, genetic research and all research related to Parkinson's more diverse ethnically, and to involve people that have been underrepresented worldwide. So it's not easy, of course, because there are a lot of structures that are difficult to change, but it's absolutely one of the goals, I'd say.
Yeah. Second row here. And then another question online following that, please.
Hi. I was quite shocked to hear that it takes about a year to do the DNA analysis. Does this impact on the start data? So if you recruit somebody tomorrow, will they be able to take part in the trial in 2026, or can they start now?
I would say that that's dependent on which sites are open. So if they get their results now, or if they already have their results.
Can't hear you very well.
I don't know if... so it's kind of dependent on when their local sites open. So as much as we could obviously send people to sites further away, there's budgets for travel and things like that, so a lot of the time people have to be sent to sites that are local to them or an hour or two away. So even if they do have their sequencing completed and they have the results now, they might not be recruited until a year's time when that site opens, but I believe that all the sites are hoping to be opened in the next few months. But also, again, the recruitment is over a 2-year rolling basis, so if someone doesn't have their sequencing results now, but they have them done in a year, they can be equally as considered for the trial as someone who currently has their results and is being, you know, contacted about sites that are open now. So at the moment, anybody who is currently getting sequenced or signed up, you know, now, they should be, you know, equally as eligible for the trial in the long run as people who already have their sequencing done, they're already in the pipeline for getting recruited to the trial. Because of this 2 year.
When you get your final results, it's going to be delayed.
Yeah, so we operate, of course, on a first-come, first-served basis because we can't hand-pick participants, and it's the time we receive the sample that counts. We sequence them in a chronological order from when we receive them. So even though your result is delayed, you're taking part in the trial or not reflects when the sample was received, if that makes sense. And we are definitely sequencing all people that need to be sequenced if they are eligible. So, of course, there are a lot of different variables as to whether or not you can take part in this particular trial, but being delayed with receiving your genetic results will not be one of those, because if the sample was sent within a certain window, you will be considered for the trial. And also, I mean, Cure Parkinson's and other organizations in the UK sponsor a lot of clinical trials, and we try to collaborate between each other to offer to everybody that is interested in taking part in a trial all the possible opportunities.
Question from online.
Yeah, so the question we have online is for Tim and Helen, and it's, what did you wish you'd known before you took part in a clinical trial?
Great question.
I don't really have any regrets for my trial at all. Was there anything else I wish I'd known? Not really, but I suppose for the good things that maybe. Oh, actually, I probably didn't know that I would have there would be so many sort of comprehensive checks and so many appointments. Although I was literally one of the last people to start exenatide before lockdown, so I had various phone appointments instead of in person. But other than that, at the start of the trial, you have a very comprehensive chat with the trial people to make sure that you understand exactly what's going on and what the pros and cons are. So, you know, I can't think of anything in a negative way.
No, I'm pretty much the same. I was probably at the beginning of the trial, and the information that came through to you was very comprehensive. They checked all the inclusion criteria, took numerous armfuls of blood, and yeah, the nurses were great. Everyone knew what they were doing. It was a real team effort. So.
Having, just to add to that question, having gone through the process now of the clinical trial, is there any feedback you would give to investigators as to how they could improve the process for the patient and participant?
I would say not so much in terms of the trial, but in the afterwards, it's just keeping people informed. And obviously with exenatide, there was quite a big delay, and it's just knowing what's going on. There was a paper produced that sort of gave, there was like a top 10 tip of all the different things within clinical trials, and keeping people informed is definitely one of them. And I would also say, in regards to clinical trials, one of the most frustrating things from my point of view, because I've signed up to lots of things, I've done every survey known to man for Parkinson's that I can find. I've had an email the other day saying, welcome to your 26th study for the Fox Foundation. I was telling to my friend, wow, 26. A lot of the time you contact people initially and you never hear anything. And I would say that from my point of view, that's one of the most frustrating things. You know, if it's yes, great. If it's no, fine. But please just tell me.
In the front row.
I just wanted to say, in response to the question about diversity and recruitment, that the EGS ASPro-PD platform, as well as having very broad inclusion criteria, has a specific recruitment target around diversity and around underrepresented groups in Parkinson's research. So the aim is, by having 40 sites geographically, and also by having remote assessments, to make it accessible to groups who can't typically take part, and also to actually ensure that we have community type initiatives to recruit people who wouldn't normally come into contact with researchers, to try and get a more representative cohort of the UK Parkinson's population as a whole, rather than the way it's often skewed in research.
Thank you for that.
Hi, thank you so much. Just a different type of question. Have you found in your research a link between a mutation of the gut and the Parkinson's disease that will affect or will have more chances to develop Parkinson's disease because you have another genetic disease linked with the gut? Thank you.
So, not personally in my research. We know that the gut is severely affected in people with Parkinson, to different extents in different people. So there are some genetic variants that are associated with the functioning of the gastrointestinal system that are associated with the risk of Parkinson's. But to my knowledge, and other people in the room might know more than me on this, there is no definitive evidence on this. But it is one that and the microbiome, which is also associated with the gut functioning, are a very hot topic in the research for Parkinson's, absolutely.
We had a question down the back there. Yep.
Thank you very much. With regard to the Ambroxol trial, do you anticipate those with the GBA variant to be prioritised?
To be quite?
To be prioritised, those with GBA variants.
So we have a flexible target of having around 50% of participants in the trial to be GBA positive. So the answer is yes and no. We try to offer it to people on a first come, first served basis, but likewise we do intermediate checks to see what is the proportion of GBA positives and GBA negatives, and then we adjust according to what is happening. So the short answer is people that have a GBA variant are more likely to be offered to take part earlier than people that don't have a variant. But people that don't have a variant will also be offered to take part on a first come, first served basis.
Super, thank you.
Another question from online?
The question specifically is, are there any issues, so if you were to be on the trial, would there be any issues sort of if you were to travel abroad during the time? But I guess this is kind of a more broad question that maybe Helen and Tim can provide some insight on in terms of when you're participating in a clinical trial, does it kind of interfere with your daily lives or, you know, plans that you have, and, you know, or is it more flexible? How sort of easy is it to participate in that manner?
Did being on a trial interfere with going on holiday?
Sorry, I missed. I could get the first bit, but I got the second bit.
It's just with regards to if travelling or activities of daily life interfere with being involved in a clinical trial.
I suppose it depends if you know how positive you are, if you want to let them do that. The trial people regarding visits were really flexible. You know, there's a window that it has to be done. But if you're away on a holiday or something like that, then they'll work around that. I actually did a trial years and years ago at my GP surgery to do with IBS, and I never did get to find out what I was taking, but for the last 2 weeks when it was finished, I still had to do daily questionnaires, and I was going to the States for my holidays. But they were so keen to get people in this trial that I had this special number to phone every day from in Florida to speak into the results, to send in the results. So I think at the end of the day, you can work around all these things. If you're not flexible and you don't really want to, then I would stick to the online stuff, because the last thing anybody wants in the trial is to start like a 2-year trial and then give up 6 months in because you know you're finding it a bind to whatever it is. So it's very much an individual thing. You know, I live down on the south coast, but we come to London all the time, so it's no big deal to me. But for other people, they might think it is, like the travelling and that. It's very personal, I think.
Yeah, I carried on working through all of mine. The clinic did an early start, so it worked great with work. Just go and get your trial off to the grindstone, as they say.
Mairead, are there any restrictions in terms of...
So we have thought about that quite a lot, and the visits are roughly every 5 months, so they're quite big gaps, and we also have a plus or minus 2-week window to allow for things like holidays.
Question from the second row.
Hiya. I've got 2 questions. First one, the 7-year rule criteria for the trial. Does that mean that it will only be licensed for people who have been diagnosed within the last 7 years? So is it restricted to people who've been diagnosed in the last 7 years?
So for the ASPro-PD study?
Yeah.
Yes, there is a eligibility criteria of 7 years since diagnosis.
Yeah, but will that be a restrictive point on if it gets formalised? I'm using the words.
Do we foresee any...
When you get it licensed, will it be licensed for people who are less than 7 years?
No, there shouldn't be any effect on that on generalization of the results. The reason for that, for picking people within 7 years, is that it's easier to see a difference in the outcomes within 7 years, looking at previous cohorts of people with Parkinson's and how the UPDRS, this scale we use as the primary outcome, went. The change is, because of statistical reasons, it's easier to see a difference if a medication actually works within the first 7 years. But of course, there is no reason to why it shouldn't then work on all people with Parkinson's, even those that had Parkinson's for longer.
The other thing I wanted to ask is what the thoughts were on stem cells, and if Cure Parkinson's are involved in any trials.
So Cure Parkinson's has been supporting the TransEuro study, which was conducted in Cambridge, which was a cell transplantation study. And the results are currently in press. They're going to be published very shortly. And that has provided the resources and the infrastructure for the STEM-PD study, which is the next generation of that work. And like a lot of the stem cell projects, the STEM-PD study, which started out as an academic-led initiative, has now been taken over by Novo Nordisk, the company that brings you the diabetes drugs, such as Wegovy. They have taken a strong interest, and they're going to take that program forward. And this is what you need with the stem cell work. You need the resources of a large pharmaceutical company. Novo Nordisk is basically the size of Denmark with all the money that they're making with the anti-obesity drugs at the moment. And they can bring the resources that are required to really power that research forward. And they're not the only pharmaceutical company. Bayer, the German pharmaceutical company, has bought the small biotech company called Blue Rock Therapeutics, and that's going phase 3 this year. It's going to be initiating recruitment very shortly. And then there is the Japanese stem cell project as well, out of Kyoto, and Mitsubishi Tuomo is taking that forward into phase 2 clinical testing. So it's quite encouraging to see these large pharmaceutical companies now acknowledging the maturity of this research and exploring how to take it forward for regulatory approval. So there's quite a lot of activity in that space that we want to watch over the next few years.
And it's also worth saying that our next research update meeting will actually be taking place in May and is on the topic of stem cells, because actually there is a consortium of researchers that are coming together, and they're actually meeting in London, and we're helping them organise all of that. And so we're taking advantage of the fact that we have this brilliant talent in the UK at that particular moment. So mid-May, so watch out for your inbox for an invitation. With Roger.
Another question from online?
Another kind of question relating to Ambroxol. Is there any kind of evidence that there's sort of like drug interactions between Ambroxol and levodopa, for instance? Can it improve it at all?
Not with any medication used to treat Parkinson's, and no major drug interactions with any other drugs, really.
We've got 5 minutes left, folks. Are there any other questions from the audience? Over on the side there, yes.
Hi. I'm interested in the effective dosage on some of these trials. It seems to me that most of the trials are a fixed set dosage. I mean, we know from our regular domestic medicines that dosage really does count. There's a big difference if you have 2 paracetamols as opposed to 1 as opposed to a half. Following on from that as well, I understand the exenatide 2 trials that differed in results had different dosages. Perhaps you comment.
There's been quite a bit of talk in the background with regards to the exenatide phase 3 and phase 2 trials having different doses. We are still waiting for the biochemical results before we have clarity on that, so I'm not sure where that has started. But with regards to the identification of, Marco, I'm going to come to you again. With regards to the identification of doses for clinical trials.
Yes, so there is the standard process for newly developed drugs is that there are, we call the ASPro phase 3 because there are phase 2 and phase 1 and preclinical studies. So the first studies are done, in vitro, so on cells, and then in animal models, and that is used to determine what dose is of a studied drug is toxic to some different animal models. And then there are phase 1 studies where the dose that are used to determine whether a certain dose that is predicted to be safe is actually safe or whether there is any toxicity in humans. Phase 2 extend on that and bring some preliminary data on efficacy, but they're mainly focused on safety. So again, they look at whether a certain dose causes adverse events, how frequent they are. And then the phase 3 is when you take the data from the preclinical studies, the phase 1 and the phase 2, and you select the dose that you think is the most appropriate, and you use it to assess efficacy of the medication. So in short, the dose is determined through preclinical and early clinical trials. And in the case of Ambroxol, it followed a similar procedure. So even though it was already available at lower doses for a chest infection, it's the studies we mentioned about treating cells with different compounds and finding Ambroxol, they gave different doses of Ambroxol to see what elicited a response in cells, and then they studied the same doses or similar in animal models, and then they moved to the phase 1 and 2 in humans. So there has been a long process to determine the dose that we believe is the most appropriate.
If there are no other questions, there is one individual on the stage who has not said anything during the Q&A. Georgie.
Hello.
I was just checking that your mic is working. Could you just remind everybody how they can take part in PD Frontline? I know you presented it before, but just very briefly.
Yeah, of course. So recognise several of you might already be part of the PD Frontline study, and if you are, thank you very much for your participation. For anyone who's not involved and would like to be involved for either the ASPro study or other clinical trial and study opportunities, you can reach out to us on our mailbox, which is pdfrontline@ucl.ac.uk, or you can come and speak to me and Millie at the end, and we'll speak to you about registering and getting involved.
I have filled my quota for getting everyone to speak. I'd like to thank everybody on the panel today. If everyone will give a round of applause.